let's talk about glomerular filtration. So, thinking back to Monday, we learned that there were three things that go into the production of urine. The first, what we're going to get ready to talk more about, is glomerular filtration, where we filter out water and a lot of other substances from our capillaries called the glomerulus. The what we call filtrate is formed that forms and is going to drain out of the corpuscle into the renal tubule, where we then perform reabsorption and secretion. And now, this was a very simplified version of this picture. The renal corpuscle is this kind of circular structure where you've got an afferent arteriole bringing blood into capillary network. Capillary network is called the glomerulus, and then the blood that does not exit leaves the corpuscle as an efferent arteriole. So we're going to start kind of talking about what's taking place in here right now. And principle of filtration is the use, or the definition of filtration is use of pressure to force fluids and solutes through a membrane. So we're going to be dealing with pressure gradients, just like we've been talking about here for the last couple of weeks. So the principle of filtration is the same in glomerular capillaries as it's in capillaries elsewhere in the body. However, the volume of fluid, I really should say, in the renal corpuscle is much larger than in other capillaries because the glomerular capillaries present a large surface area available for filtration because they're very long and extensive. Our filtration membrane is very thin and porous. Remember, those capillaries were described as being fenestrated, and that means that they've got all those little tiny holes in them. They're about 50 times leakier than capillaries in most other tissues because of those large fenestrations. And then the pressure inside of the glomerular capillaries is really high. And that's because, and actually, if I go back to this page, it's kind of hard to show, but look at the size of the afferent arteriole compared to the efferent arteriole. Okay. This is, showing it a little bit more, but the width across the afferent arteriole, the lumen, in other words, is bigger here than it is here. Okay. So it's kind of like this blood vessel is kinked a little bit. It's constricted just because it's not as open or more, it's just smaller. So the blood in here is under a lot higher pressure. Okay, and that's going to try to drive that fluid out of the glomerular capillaries. Okay. So glomerular capillary blood pressure is high due to the efferent arteriole being smaller in diameter than the afferent arteriole, causing resistance to the outflow of blood from the glomerulus to be high. So we're talking pressures. There's actually three pressures that affect glomerular filtration, or that we could say glomerular filtration is dependent upon them. The first is that pressure inside the glomerular capillaries that I was just talking about. This is called glomerular blood hydrostatic pressure, GBH-pre, okay, and it's one that will promote filtration by forcing the water and solutes out into the capsular space through the filtration membrane that we talked about last time. The other two are actually opposing that. Looking at our figure, we show some arrows pointing in. These purple arrows represent the capsular hydrostatic pressure, or CHP. And this is the pressure exerted against the filtration membrane by fluid already inside the capsular space and inside the renal tubule. Okay, so there's fluid in here, which is going to oppose the movement of more fluid out. Okay. Now, the third pressure is what we call the BCOP, blood colloid osmotic pressure, which is pressure due to the presence of proteins like albumins, globulins, and fibrinogen in our plasma. Both CHP and BCOP oppose filtration. So, in other words, we have to do a calculation to see if filtration is going to happen. We call this what we calculate using the equation net filtration pressure, the total pressure promoting filtration. So it's the big number, the first one we talked about, GBHP minus CHP minus BCOP. This one, remember, promotes filtration. These two inhibit it, so we take the one that promotes it minus the other two. Yeah. We actually talked about this a little bit before, but here's the basics of it. Okay. When you do the calculation using the numbers they provide, 55, 15, and 30, 55 minus 15 minus 30, the NFP calculation is a positive 10 millimeters of mercury. Okay. Simply put, and it might not be a bad idea to write this down, a positive NFP. So if this number is above 0, we're going to form filtrate. Okay. So positive NFP equals filtration. If you want to just write it down that way, it's kind of a simple way to think of it. If this number is negative, though, like say it's negative 10 instead of positive 10, a negative NFP inhibits filtration. So positive NFP promotes filtration, negative NFP inhibits it. Now, are you going to have to know, the specific pressures, know the calculation? No. Definitely know, what NFP is. And then, like I said, know that positive NFP promotes filtration and negative NFP inhibits it. Now, we need to know this, and it's important to know this because filtration, or NFP, excuse me, affects what we call filtration rate. Okay, GFR, short for glomerular filtration, refers to the amount of filtrate formed in all the renal corpuscles of both kidneys each minute. So this is a measure of how much fluid we're pulling out of the blood along with the other stuff, because it's not just water. It's all those little tiny ions and other things that are small enough to pass through the filtration membrane. That's per minute or each minute. Okay. This is an average of 105 mils per minute in women and 125 mils per minute in men. Here's some crazy statistics. That adds up to 180 liters or about 45 gallons of filtrate formed per day. Thinking of like a pop bottle. Okay, that's a 2-liter pop bottle if you're thinking of it that way. Okay, 180 divided by 2 is 90. That'd be 90 of those pulled out of the blood every day, or 45 gallons, that'd be 45 milk cartons. That's insane. Obviously, we're not peeing nearly that much, in which I say right away, most of the glomerular filtrate must be returned to the blood and recycled. If we were peeing out that much water, we would have to be just drinking water all the time, all the time. Okay. Homeostasis of our body fluids requires that our kidneys maintain a relatively constant GFR. If it's too high, needed substances may pass so quickly through the renal tubules that they may be lost in our urine. If it's too low, nearly all the filtrate will be reabsorbed, meaning that it will be returned to the blood, and certain waste products may not be adequately excreted. As I was just saying, as I flip from the previous slide to this slide, it's important to understand NFP because GFR is directly related to those pressures that determine NFP. In other words, any change in that filtration pressure affects GFR. Okay. There are 3 mechanisms that control our filtration rate by either adjusting the blood flow into and out of the glomerulus or by altering the capillary surface area available for filtration. Okay. The first is called renal autoregulation. Thinking back to our blood vessels lecture, we learned autoregulation was the way that we regulate blood pressure within our capillary beds. Remember, auto means self. So this is a way that our kidneys can self-regulate, in this case, GFR. Neural regulation refers to the nervous system, and we'll be talking sympathetic, parasympathetic again. And then hormones affect our filtration rate. Let's talk about autoregulation here first, though. Okay. Renal autoregulation is our ability of our nephrons to adjust their own blood flow in GFR without external, in other words, nervous or hormonal control. So essentially allows the nephrons within our kidneys to control their own GFR without outside, outside influence. There's a word for it. This allows them to maintain a stable GFR in spite of changes in arterial blood pressure. Two mechanisms of renal autoregulation exist, what we call a myogenic mechanism and tubuloglomerular feedback. First myogenic mechanism occurs when stretching triggers contraction of the smooth muscle cells in the walls of the afferent arterioles. Okay. Looking at the figure here on the left side of the page, this is the myogenic mechanism. I'll kind of bounce back and forth with this figure and the information. Now, let's just go to this picture. So we're going to be talking about adjusting the width of the afferent arteriole. In other words, adjusting the lumen of the afferent arteriole, which affects the amount of blood that's going to flow into the corpuscle. Okay. So when stretching of the afferent arteriole occurs, it triggers contraction of the smooth muscle cells in the walls of that afferent arterioles. As blood pressure rises, GFR also rises because renal blood flow increases. Okay, so if there's a high blood pressure, there's a high force of that blood into our kidneys, which means a lot of blood is going to flow into those afferent arterioles, and we're going to start filtering out fluid from the blood. So as blood pressure goes up, GFR also goes up. However, elevated blood pressure stretches the walls of the afferent arterioles, and in response, the smooth muscle fibers in the wall of the afferent arterioles will contract, which narrows its lumen. So going back to the figure, if these things are stretched because of a surge of blood, when blood pressure is high, we're going to be forming a lot of filtrate. Well, we don't want to just form way too much. We want it to be stable. So our afferent arterioles stretch, but in response, the muscles that line them contract. And so we vasoconstrict essentially those afferent arterioles. And as a result, GFR reduces to its previous level. Okay. That's essentially what we're showing in this segment of our figure. Okay. Arterial pressures go up, causing GFR to go up. The afferent arterioles stretch. Okay, and now it goes into a little bit more detail, essentially what happens. But ultimately, we constrict the afferent arterioles so blood flow goes down in them and GFR goes down and thus restores our normal GFR. This is an example of negative feedback. Now, the opposite occurs when arterial blood pressure is low and those smooth muscle cells are stretched less. Okay, so if blood pressure is low, not as much flow is going to be coming into them because there's not as much force of blood flow. So there's not as much stretch, but our afferent arterioles respond by dilating, and that means more flow will come in. And because GFR is going low, we want to increase it to return it to normal. Okay. The myogenic mechanism is going to help normalize renal blood flow, so kidney blood flow and GFR within seconds after a change in blood pressure occurs. So it's very, very rapid, very, very, good at what it does. The tubuloglomerular feedback is a feedback loop involving the macula densa of the juxtaglomerular apparatus. If I go back to our figure of the nephron, remember a point in the distal convoluted tubule right here, it passes by that afferent arteriole. Okay? And here we show those cells of the distal convoluted tubule can communicate with these cells of the afferent arteriole. And they provide feedback, which we literally call tubuloglomerular feedback. So as GFR increases, so we're making a lot of filtrate, less solutes are reabsorbed in the proximal convoluted tubule and the loop of Henle. So essentially, we're making a lot of filtrate, but we're not returning stuff back to the blood that we need to. The cells of the macula densa detect those chemicals and in response secrete chemical messengers, causing the afferent arteriole to vasoconstrict, and chemicals that inhibit afferent arterial dilation. So the afferent arteriole is not going to dilate, it constricts, less blood flows into the glomerulus, and GFR in that same nephron decreases. Okay. So going back to this figure again, if we're tasting, and I'm using that very loosely, okay, these cells are realizing that we're not returning nutrients back to the blood, we're going to end up peeing those out. Okay. These cells are going to tell the afferent arteriole to close up. Okay. Slow down the flow of blood into the glomerulus so that the flow of filtrate is much slower. Our rate is lower, which means we can return those things back to the blood that we need to. Now, the opposite occurs essentially when blood pressure falls, causing GFR to be lower than normal. The sequence, like I said, is opposite, although to a lesser degree. Tubulo-glomerular feedback operates more slowly than that myogenic mechanism that we just talked about. Okay, looking at this figure on the right side of the page is an illustration of that feedback loop for tubulo-glomerular feedback, showing you what happens when GFR goes up. Those macula-densa cells communicate with the afferent arteriole, causing the afferent arteriole to, in this case, decrease its secretion of nitric oxide, which means the afferent arteriole constricts, less blood flows in, GFR goes down, and we return back to a normal controlled condition of GFR. Okay, so that's how our kidneys can self-regulate. Okay. The kidneys are subject to outside influence. Thinking back to the middle part of the semester when we discussed sympathetic, parasympathetic, really this should be pretty common sense based on what we know. Okay, we know sympathetic nervous system produces a fight or flight response, and this sympathetic nervous system will trigger the release of norepinephrine that causes vasoconstriction of arterioles in the kidneys, especially the afferent arterioles. Just from what we know about a fight or flight response, I've talked about the fact that when we are in a fight or flight response, our body should not be concerned with, kind of two major things: digesting our food and making urine. Okay. There's other, obviously, things, but those are kind of two simple things. So if we know that our sympathetic nervous system is active, we should know that we aren't going to be doing much for filtration. Okay. Now at rest, when there's moderately low sympathetic stimulation, in other words, our parasympathetic nervous system is active, both the afferent arteriole are dilated, and therefore renal autoregulation of GFR is more important, or it prevails. But with moderate sympathetic stimulation, both the afferent and efferent arterioles constrict to the same degree, which causes a slight decrease in GFR. Even greater sympathetic stimulation, like during exercise or like during hemorrhage when a person is bleeding, this causes constriction of the afferent arterioles more than the efferent arteriole. And remember, the afferent arteriole brings blood into the corpuscle, efferent leaves or allows the blood to leave it. As a result, blood flow into the glomerular capillaries is greatly decreased, causing GFR to drop. Okay. Now, this is important because two things. We're allowing blood to go where it needs to go, our skeletal muscles, our cardiac muscle, but we're also not pulling stuff out of the blood like the water, which, we, as we said, pull out a lot every minute. Okay, so if we're not filtering out water from the blood, our blood volume will go up. Okay, and so blood volume's up. That's great, which means we're going to have a better ability to deliver the nutrients and the energy and that kind of stuff to those working muscles. Okay, so a reduced urine output helps to conserve blood volume. Greater blood flow then is able to go to our other body tissues, things like our skeletal muscle and our cardiac muscle that are working. So hopefully pretty straightforward. We know sympathetic nervous system, fight or flight response, going to kind of shut down activity of the kidneys. Okay. Whereas parasympathetic is going to promote the formation of urine, is going to enhance filtration rate, or the formation of filtrate, I should say. Okay, several hormones influence our kidneys. Two kind of major hormones influence GFR: angiotensin II and atrial natriuretic peptide. If we think back to our lecture on the blood vessels, we talked about the hormones here on this page, and how they related to blood pressure. Okay, we learned in that lecture that atrial natriuretic peptide is secreted by cells of our heart's atria, the two upper chambers. Not only does it influence blood pressure, but it's going to influence GFR. Okay, we learned that atrial natriuretic peptide is secreted in response to basically excess blood. Okay, high blood volume. So that's going to affect blood pressure. What it's going to do in the case of our kidneys is going to increase the capillary surface area available for filtration by causing relaxation of those glomerular mesenchymal cells, the cells that are found kind of sprinkled in between the capillaries. So we have more surface area available for filtration, so our GFR will go up. We'll filter more stuff out of the blood, including that water, which makes the blood volume go down. Okay. And thus, we're going to return our blood volume to normal, and it will not need to release this hormone anymore. The renin-angiotensin-aldosterone mechanism is what we release, and I shouldn't say release because it's a series of things. It involves enzyme called renin from our kidneys. Angiotensin and aldosterone are also involved, obviously, hence the name. We learned that this whole mechanism is going to trigger increases in blood pressure, and it's released when or these things are active when our blood pressure drops, as it states right there. Okay. It involves sympathetic nervous system signaling cells in our juxtaglomerular apparatus to secrete the enzyme renin, which interacts with a plasma protein angiotensinogen, causing it to release a chain of amino acids called angiotensin 1, which is then converted to angiotensin 2 in our lungs. Okay. Angiotensin 2 is the actual hormone that promotes a lot of different things. Okay. It stimulates widespread vasoconstriction, which increases blood pressure. So causes blood pressure to go up when blood pressure was down. It constricts within the nephron both the afferent and efferent arterioles, reducing GFR. Reducing GFR means that we lose less water, meaning that our blood volume goes up. Any increase in blood volume, we learned, increases blood pressure. So bullet number 2 here tells us that blood pressure is going to go up as well. Stimulates sodium chloride or salt and water reabsorption, meaning that we pull more salt and water back into the blood, thus increasing blood volume, which obviously is going to affect blood pressure, as I just said. So this is going to promote increases in blood pressure as well. Stimulates the adrenal cortex to release aldosterone. Aldosterone is a third hormone that I didn't list at the top, but it's involved here. Aldosterone, we learned, is our salt retaining hormone. So it's going to promote sodium retention, but also, in doing so, promote water retention, which causes an increase in blood volume, which thus causes an increase in blood pressure. Also stimulates another hormone to be released, ADH, from our posterior pituitary, antidiuretic hormone, helps us return water to the body, so it promotes water reabsorption, thus causing an increase in blood volume. And just the same, any increase in blood volume increases blood pressure. And then it also just makes us thirsty and encourages us to drink water, which obviously is going to increase our water, thus increasing our blood volume and thus increasing our blood pressure. So, very complicated series of events. A lot of different things go into it. I'm not going to walk through it all, but essentially, kind of increases blood volume by way of decreasing filtration rate, and through a series of events, it increases your blood pressure as well, which should make sense because the whole kind of important reason why we activate this system was a decrease in blood pressure. Okay, table 26.2 just is a summarization of those 3 different mechanisms that regulate GFR: autoregulation, neuroregulation, and hormonal regulation. Okay, on to the next part of our urine formation process. We're going to talk about tubular reabsorption, and we'll get into tubular secretion here in a little bit. Now, thinking back, I'm going to backtrack to our picture of the nephron. All we've talked about at this point is what's taking place right in here. Okay, along with the afferent arteriole, the glomerulus, and the efferent arteriole, where we first form filtrate and collect that filtrate and then drain it into our renal tubule. From here on out, we're going to be talking about returning stuff from that filtrate from the tubule into the blood. Returning it is called reabsorption. Okay. And the opposite of that, moving stuff from the blood into the tubule, is what we call secretion. And so we'll be talking about secretion here as well. Defining tubular reabsorption, again, we've already defined it once, but this process of reclaiming water and solutes from our tubular fluid, returning them to the blood. It occurs in the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and we finalize it in our collecting ducts. Okay, we said we filter out like 45 gallons, I think that was the statistic, of water and other stuff of what we call filtrate per day. And normally about 99% of that water is absorbed or reabsorbed in those structures on our proximal convoluted tubule, makes the largest contribution. So we pull out that water and stuff from the blood and our corpuscle, and then we give most of it right back in the proximal convoluted tubule. Now, most of our water and solutes are recovered by our blood via two routes, what we call a transcellular route and a paracellular route. Trans is a prefix that always means like across or through. So this is where substances pass through the cytoplasm and out the basolateral membrane of the epithelial cells that line the tube. We call this transcellular reabsorption. Paracellular is where the cells where the substances are passing between the cells that line the tube. We call this process paracellular reabsorption. Here we can just show it. It's a lot easier to show some figures to make it make sense. So at the top of the page here on the next slide, we show sodium paracellular and sodium transcellular reabsorption. Both processes are going to get sodium from the tubule lumen, so inside the tubule, back into the bloodstream. The difference, transcellular, we're going through the cells. So we pull it in on one side of the cell, crosses through the cell, and then out the other side. Paracellular just allows the sodium, in this case, to escape kind of between the two cells. So that's really the difference. Now, sodium is super important. It is going to help move other stuff, okay? Which is why I have it listed here as, or I have it typed up as reabsorption is primarily coupled to movements of sodium ions, which are actively being transported out of the tubular cell and into the surrounding interstitial fluid and blood plasma by what we call a sodium-potassium ATPase pump. Okay, the sodium-potassium pump requires energy. We're showing it right in here, pumping sodium from the cells into the interstitial space. Okay, now by pumping sodium out of the cell is never going to have much sodium inside. Okay, remember, active transport pumps things against their gradient from low to high. So we're pumping what we already have low of out, which makes the amount of sodium inside always really low. Okay, which means sodium is always going to be trying to come into the cell from the tubular lumen. Okay. Now, in doing so, sodium can come into the cell by way of a sodium glucose symporter, which allows glucose to hitch a ride. So two sodium ions can come into the cell down their concentration gradient, and glucose can piggyback ride along with it. And once glucose enters the cell, then it can just use a glucose transporter to get out of the cell into the interstitial space, where it can then jump back into our bloodstream. And so it's kind of complicated, but if you kind of just think of it, it's hopefully not too terribly sophisticated in how it all works. Now we're going to do the opposite for hydrogen ion that we did with glucose. Instead of pulling glucose in, or instead of pulling hydrogen ions into the cell, we're going to push them out of the cell into the tubule lumen. And this is done by way of what's called a sodium hydrogen ion antiporter. Symporters move things together. Antiporters move them in the opposite direction. Okay. Now, where does this hydrogen ion come from? Well, it's coming from that same equation we've been talking about for the last few weeks. Carbon dioxide and water can combine thanks to the enzyme carbonic anhydrase, forming that acid that we previously learned about called carbonic acid, which then breaks apart, forming hydrogen ions and bicarbonate. Most of the bicarb is reabsorbed into our bloodstream, where it can then act as an important pH buffer. The hydrogen ions that are formed are pushed out into the tubule lumen, which then is going to end up making the fluid inside the tubule lumen very acidic. Okay, not super acidic, but slightly acidic. Well, that's great because this is going to allow us to get hydrogen ions out of our body and thus help to maintain our pH. Okay. Now, after hydrogen ion is secreted in the lumen of the proximal convoluted tubule, it reacts with filtered bicarbonate, forming carbonic acid again. So essentially, we're kind of seeing the same thing take place in reverse that we talked about back up here. Okay. So the carbonic acid dissociates, forming CO2 and water. So there's where those hydrogen ions end up going out of our body in the form of water, thus allowing us to remove them from the body and maintaining long-term pH in our body. The CO2 that was formed, we don't pee out the CO2 because the CO2 returns into those cells to then go through the process again. So we're just kind of doing the same thing on both sides, just kind of the opposite direction, but we're forming water, which enables us to get rid of those hydrogen ions, which is super important. For solutes, things like chloride and magnesium, potassium, calcium, etc., things dissolved in the lumen, they're going to help promote osmosis of water. Okay, remember, and I've talked about this before, water is always going to chase after things like salt and other ions. So when, let's just say sodium, or it's going to be chloride, potassium enter the cell, water is going to try to go into that cell as well. When they leave the cell and they enter the interstitial space, water is going to leave the cell and enter the interstitial space. When they enter the blood, water is going to chase after it. Okay. Now, water can move through the cell through what we call transcellular routes. It can go between the cells via paracellular routes. Okay. So, the essential goal is to get water back to the blood, but in doing so, we're also returning a lot of those various ions and other solutes that we filtered out back in our renal corpuscle. Now, that was all in the proximal convoluted tubule. We haven't got very far because there's still 3 more segments to talk about. The loop of Henle, the distal convoluted tubule, and then the collecting duct. Let's talk about the loop of Henle. Okay. We're going to be performing both reabsorption and secretion here. It's kind of a complicated process, so let me read through and explain what's taking place. Okay, because the proximal convoluted tubule reabsorbed about 65% of the filtered water, fluid that enters the loop of Henle, really should say not with, but has a very different chemical composition than it was entering the proximal convoluted tubule. So that sentence basically means, we returned a lot of that stuff back to the blood already. So the fluid inside of the loop of Henle is a lot different than it was back up here in the proximal convoluted tubule. We're going to continue reabsorbing water, about 15% of that water that we filtered out, along with those various ions that we've been talking about. But for the first time, water is not going to be coupled to the movement of those solutes because part of the loop is relatively impermeable to water. Impermeable means that it's not going to be able to pass through. Okay, water is not able to escape the tubule in what we call the thick segments. And the other day we talked about the fact that the loop of Henle has some thinner segments. It's got some thicker segments. Okay. The thin segments are absolutely permeable to water. Water is easily going to pass out of those thin segments, but the thick segments, water is not able to pass through, okay, and out. Okay. Now, tubular reabsorption and secretion in our loop of Henle occurs via what we call a countercurrent multiplier mechanism. Very complicated sounding name, but it refers to an interaction between the descending and ascending loops of Henle. So, in other words, what's taking place here and here is influencing each other. Okay, more so, what's taking place here, is influencing what's taking place here. Okay, oops, wrong way. All right, so let's talk about what's taking place. Water is reabsorbed from the tubule fluid via osmosis in the descending limb. And this is accomplished because the interstitial fluid of the renal medulla is very hypertonic and thus has a very high osmotic pressure. Okay. These numbers that you see kind of periodically throughout this figure are describing essentially the drive for stuff to leave the blood. In other words, mainly we're talking about water. Osmotic gradient is what we're focusing on to promote osmosis, movement of water. The bigger the number, the higher the drive for water to leave the tubule. Okay. Notice the bigger numbers are deeper down in our figure. This is all down here below this dotted line, the renal medulla. Okay. The renal medulla, I mentioned the other day, is really salty because actually what we're doing in the ascending limb is pumping out sodium and chloride. Sodium and chloride, remember, that's just table salt. Okay. This fluid that's in the renal medulla is going to be super salty as a result. Okay. Salt water is described as a hypertonic solution. We learned about hypertonic, hypotonic, isotonic stuff at the beginning of the semester. Okay. We learned that if you drink salt water, for example, your body is going to get dehydrated because your blood becomes very hypertonic. Water is going to try to go places that are really salty. In other words, like in this case, our renal medulla. Okay. By pumping out salt here, this fluid that surrounds the tubule, the interstitial space, the interstitial fluid is super salty, which draws the water out of the descending limb. Okay. Now, it's not shown in this illustration here, but surrounding the loop of Henle are blood vessels called vasa recta that pick that water up immediately. So when the water escapes the tubule, it's going to go directly into the bloodstream. And that means the fluid, this interstitial fluid, never gets diluted. Okay. Even though water's leaving to go where it's salty, the water is picked up by those blood vessels all the time. Here, actually, you can see those vasa recta blood vessels that are picking up that water from the blood, or picking up that water and returning it to the blood almost instantly after it's leaving the descending limb. Okay. Now, on top of that, we're also picking up what's called urea from the interstitial fluid in the descending limb. Okay. So you can see that right here with the white arrows. So urea is actually being secreted. Remember, tubular secretion is moving stuff into the tubule, whereas water is being reabsorbed and it's moving out of the tubule. Okay. We're also reabsorbing salt, okay? And that's taking place in the ascending limb, which once again makes the renal medulla super salty, super hypertonic. But inside the tubule, as the salt's leaving the tubule, it's becoming pretty hypotonic, which is basically the opposite. Not basically, it is the opposite of hypertonic. Hypotonic means it's a lot less solutes. It's more water. It's diluted anyway. In other words, and that sodium chloride is moved into the surrounding interstitial fluid of the renal medulla due to, and this is what's shown here on this page, active pumping in the thick segments Active pumping means ATP is required to move the sodium, the chloride, the potassium across the cell and into that interstitial fluid of the renal medulla. Now, that urea that we are picking up in the thin segments, so we're secreting it, is actually being reabsorbed in the next door collecting duct. Okay? So that urea gets picked up, and then we eventually travel the filtrate down into the collecting duct where we're almost at the point where we can call it urine. At this point, we actually pull the urea back out of the collecting duct into the interstitial space where it's then picked up. So we call this urea recycling. We're reabsorbing the collecting ducts. We secrete it in the tubular fluid in the descending loop and the thin segment of the ascending loop, which is done because it promotes that water reabsorption, kind of like what the salt did. So the whole reason we call it a countercurrent multiplier mechanism is because what's taking place in one limb is multiplying and affecting the other limb. Okay. Super complicated, and I tried to kind of dumb it down as best I can, but it's pretty complicated stuff. So main thing is make sure countercurrent multiplier mechanisms taking place in the loop of Henle. What's happening with water? It's being reabsorbed in the descending limb. Salt is being pumped out in the ascending limb, so salt's being reabsorbed. We're performing urea recycling. When we're pulling it out of the tubule in the collecting duct, we're putting it back in the tubule in the loop of Henle. Now it is 10:50, so we are going to stop here. This is just fine because we are at a point where we can pick up next week talking about reabsorption and secretion in the distal convoluted tubule in the collecting duct. So